Okay, so when I was asked to give a community effort, I started to realize that the gut community wasn't that well connected, at least, or maybe I was not so well connected to the network because I'm a new starter in this part. So what I did is I asked all the major players for a couple of slides, and you will see that the differences in the slides are interesting, from very simple ones to extremely data-dense slides, which I could not present. So I'll start off with the work from the Fu Chao lab a couple of months ago in Nature Cell Biology, where they started to look at developmental aspects of intestine, starting to compare very early stages and then into maturation into more mature stages. And essentially, I think this is the very first map, or at least the first one I could find, on the intestine development. And basically, I put some of the highlights down here that came out of that story, of that publication, which is in particular the involvement of signaling pathways in the small intestine development. They reported on the molecular mechanisms for digestion and absorption of proteins, and again in the small intestine, and some critical features of the cells in the fetal gut. I restricted it to human because a recent publication on the mouse work is also dissecting the different zones here, the Iskovich study. But I thought for the human cell atlas, I stick to human. The second one is a slide from Sarah, which is a very complicated slide. It basically shows where we find the intestine, and then the number of projects that they're starting. So basically going from biopsies and disease, transplantation donors, and post-mortem, and the focus will be on celiac, ulceritis, colitis, and IBD. And the material here and the tissue comes from the Cambridge biorepository that was already mentioned. Then moving into the colon atlas, and this is a more complicated slide. Thank you very much, Aviv. And it came in last night when I was basically asleep from my jet lags. So I am essentially looking at it with you for more or less the same time. And not completely, I glanced at it this morning, but that was it. So basically what we're seeing here are I keep on doing the wrong one here, looking at the epithelial cells, the immune cells. We come back to that later, and maybe we should have some exchange of data here, as well as at the stromal inflammatory interaction of the monocytes with T cells, I guess, here, and high resolution. And this is now mouse. I would have removed it if I would have seen it was mouse. And the human cell here, the genetics, the interface of genetics and single cell data that we're doing. Then the work from the Clavers lab, and basically we all know the work that started very intensely many years ago on the colon organoids, and the important information we got out of there. Now, basically, they are analyzing full tissue and the different cell types in the tissue. And we see here, in particular, interesting all the T and the B cells. And the comparison, and that's, I think, a very useful comparison of normal mucosa and colorectal cancer. And you see, for example, here, complete new sets of of cells or clusters occurring, which are the CD8 T cells. And I will come back in the next slide on that as well. So basically, this is early times. It's a small number of cells, but the number is increasing now, and also the in-depth analysis of the different T and B subsets and myeloid cells is ongoing. That brings me to a slide that I know a bit better, which is our own work, basically looking at celiac disease, and that's a collaboration with Frode Jansen in Oslo. We've been, at this point, been able to look at 10 different patient samples, and since the time is running, I'll just give you 2 examples here, what happens in the CD4 T cells, and in blue is marked the healthy ones, and in brownish red, the celiac. And you see that they start off in the CD4 T from a progenitor group, and then there's a bifurcation at some point into a healthy and a disease route. And similarly, if we look at the myeloid cells, we see again the trajectory of the myeloid cells. The ones in celiac are restricting to the very early stages of after infiltration of the monocytes and then differentiation into macrophages. And in the healthy ones, we see fully matured or more matured samples that we do not see in the myeloid of the celiac patients. Last slide is again a slightly more complicated one. Basically, it's the ongoing activities in the pediatric gastroenterology of Edward Nieuwenhuis. Basically, what they are trying to do is understand the mechanisms of epithelial and microbe interaction at different levels, at the level of organoid model systems, and looking at the response in a single cell analysis upon exposure. Similarly, identify and look at what happens at the villi, as well as in the more cell culture system to look at what the interactions are and what the consequences of these are. So with that, I'll stop here, and if there's a question, happy to answer.